Hello everyone, my name is Shreya. Today my topic is eukaryotic transcription. Now let's see what is transcription. Transcription is a process in which a gene's DNA sequence is copied to make an RNA molecule. RNA polymerase is the main transcription enzyme. Transcription begins when RNA polymerase binds to a promoter sequence near the beginning of a gene. RNA polymerase uses one of the DNA strand as a template to make a new complementary DNA molecule. Now let's talk about what are the enzymes involved in transcription. Three types of enzymes are involved in transcription. Those are RNA polymerase 1, which is also known as RNA pol 1. It is located in nucleus and transcribes the 28S, 18S, and 5.8S rRNA genes. The second one is RNA polymerase 2, known as RNA pol 2, located in nucleoplasm and transcribes protein coding genes to yield pre-mRNA. Third one is RNA polymerase 3, known as RNA pol 3, is also located in nucleoplasm. It transcribes the gene for the tRNA, 5S rRNA, U6 snRNA, and 7S rRNA. Steps involved in eukaryotic transcriptions. The first one is initiation. Each gene has its own promoter, and a promoter contains DNA sequence that let RNA polymerase to attach to the DNA. A typical bacterial promoter contains -10 to -35 DNA sequence. RNA polymerase recognizes the sequence to bind to that. DNA opening occurs at the -10 element, where the strands are easy to separate due to many As and Ts. The next step is elongation. Once RNA polymerase is in the position of elongation starts. It is basically the stage when the RNA strand gets longer. During elongation, RNA polymerase works along one strand of DNA known as template strand in 3' to 5' direction. The RNA transcript is nearly identical to the non-template strand of DNA. RNA strands have the base uracil in the place of thymine as well as a slightly different sugar in the nucleotide. Termination, there are two termination strategies found in the bacteria, Rho dependent and other one is Rho independent. Rho dependent, here the RNA contains a binding site for the protein called Rho factor. It binds to this sequence and starts climbing up the transcript towards RNA polymerase. When it catches up with the polymerase at transcription bubble, Rho pulls the RNA transcript and the template DNA strand apart, releasing the RNA molecule and ending the transcription. Rho independent, the RNA transcribed from this region folds back on itself, and the complementary C and G nucleotides bind together. In the terminator, the hairpin is followed by a stretch of U nucleotide in the RNA. The complementary UA region of the RNA transcript forms only a weak interaction with the template DNA. This, coupled with the stalled polymerase, produces enough instability for the enzyme to fall off, liberate the new RNA transcript. RNA processing. Cleavage. Larger RNA precursors are cleaved from the smaller RNAs. Capping and tailing. Initially at 5' end, a cap and a tail of poly A at 3' end are added. Splicing. The eukaryotic primary mRNAs are made up of two off segments, non-coding introns, and other one is the coding exons. Nucleotide modifications. They are most common in tRNA methylase and deaminase fashion. Significance: transcription of DNA is the method for regulating gene expression. It occurs in the preparation for and it's necessary for the protein translation. These are the reference. Thank you so much.